Unsupervised analysis methods can help us understand structure, distance, and similarity in our data. Here I have a dataset of gene expression that has a number of genes, 6,918, and a number of samples, 52. These samples can be organized into different groups, and inside each gene, I have its gene expression values in FPKM values. Now, if I would like to understand the relationship between two genes, I can simply make a scatter plot, and then I will know the relationship between the expression values of one vector of a gene and another vector of another gene. I can do the same for two particular samples. Just looking at two samples, I can create another scatter plot and see the relationship between them. I would expect similar genes to have similar expression and similar samples that fall under the same group to also have a similar expression pattern. But when I come to a data set that has thousands of genes across tens or hundreds of samples, it's really difficult to get a grasp or a snapshot of what that really means. Unsupervised analysis of big data can help us identify patterns, relationships, and important structures inside of our dataset. To do that, we can go to the unsupervised analysis section and upload the data that I was just showing. Since our data is annotated by group, I'm going to press on Continue and select Start, PCA library. Here I have a number of parameters that I don't really have to change, but what they mean is the number of output principal components that I will get in my chart, whether I want to scale or not the data that I am putting in, do I want to center the data, and whether the data has to be transposed. In other words, do I want to look at the relationship between samples or objects in my data or genes, in other words, features or rows in my data. And so I'm going to say yes to analyze the relationship between samples which are annotated in my dataset. Now I will click on End. After I click on End, I'm going to give it a second to create the pipeline. I will name it, save it, and now I can run this pipeline on the cluster. It might take a little bit for the dataset to process depending on the size of the dataset and the number of users running this analysis at the same time on the shared cluster. As soon as the pipeline has completed, I can click on Download Pipeline Output Files and find some results that include text files, PDF files, and also a packaged R script that I can use. Let's take a look at the plots in PDF. Remember that principal component analysis is a dimensionality reduction technique. So what we see is the whole dataset reduced to just two dimensions, principal component 1 and principal component 2. Inside these two dimensions, we will find dots that represent objects or samples in the dataset. The distances that we observe represent a similarity between samples. So when they are closer together, they are more alike. And when they are far apart, they are more different from each other. This difference is based on variance in the sample data itself. So whenever we see some two dots closer together, that means that they are not as varied compared to the rest of the data set. And that's what you see here in these percentages next to PC1 and PC2. This is the total variance represented by each component. So the variance of PC1 and PC2 together, or the full plot, is 12.62 plus 18.08, or about 30%. Now in the next two PDF plots, you will find that the same data could be represented on PC1 and PC3, together representing about 25% of the data, and PC3 and PC2. Some of these combinations of principal components show a much better separation between samples based on group, based on the annotation that we provided to the analysis method. So here you can see basal, cloud and low, luminal, and normal-like. Cloud and low are fairly separate from all the rest, as well as all of the other colors are clearly separated within this principal component space. Analyzing the data in such a way can lead us to generate a novel hypothesis. The hypothesis might be that all the groups are different, and the biggest difference is between the luminal and all of the other samples combined. Given this insight, we can now test our hypothesis, developing a supervised analysis method that would be looking at the differences between these two groups.